Vamos a comenzar. Buenas tardes a todos. Muchísimas gracias por encontrarse con nosotros el día de hoy. Me va a permitir dar como medidas generales. Bueno, sabemos que no, salvo el ponente, se prefiere que no se ingieran alimentos y vamos a estar transmitiendo en YouTube. Entonces, les pediríamos que pudiéramos hacer al finalizar la ponencia, pues una sesión de preguntas tanto en línea como para los presentes que se encuentran aquí el día de hoy. Me voy a permitir presentar a la doctora Hilda Martínez Coria. Ella es profesora de nuestro departamento, del Departamento de Fisiología, quien en su momento nos hizo la cordial invitación del doctor Suhail Rasul.
Voy a hacer una pregunta. La gente va a estar haciendo, ¿verdad? El público.
¿Quién está hablando? Ok, bueno. Nos hizo la cordial invitación de que pudiera venir el doctor Suhail Rasul y evidentemente en la coordinación pues aceptamos con todo gusto. Dr. Suhail, thank you to be with us today, and for the department, it's a great honor to have you with us. Doctora, muchísimas gracias.
However, we have a very great talk today about the development of new therapy for treating neurological disorders. So please give a warm welcome to Dr. Suhail Rasul, who is Director of Neurology and Neurodegenerative Disease Research Division of True Binding Company from Foster, California. So thank you very much for coming, and to Dr. Rasul for giving us your time.
Yeah. Thank you. It's working. Working? I hope so. Okay. First of all, good afternoon, everybody. Thank you very much I really appreciate, first of all, Dr. Hilda Martinez Coria and Victor for inviting me to give a talk about what we have done. We actually have planned this sessions to visit Mexico to give a talk to the students. The students, it's okay now? To give the students, to give the talk, what we are doing and what we have done before. So in 2007, we started working at UC Irvine. That's the journey started. So before that, I did my medical school and PhD from Only Institute. So we started looking at working on development of therapeutic modalities for various neurodegenerative diseases. So this is a basic thing that if you see how Alzheimer's was discovered, how it came into existence, it was Dr. Elias Alzheimer's in 1906 who discovered Alzheimer's illness when he was treating a patient who was 51-year-old named Augusta D. And since then, millions of people are suffering from Alzheimer's disease. So this is so far as far as the AD statistics is concerned, as far as the United States is concerned. It's one of the most common cause of dementia among people age 65 and older. And as it's estimated, 4.5 million people have now Alzheimer's disease. And for every age group beyond 65, the percentage of the people with AD doubles. So it not only have a health issues, it also has an economic impact. If you see in the United States, almost $100 billion are using to care for those Alzheimer's patients. And by 2050, if we don't have a cure for Alzheimer's disease, so we think that it's 13.2 million older Americans are. That one?
Yes.
Oh, I don't know. Okay. But you are listening. Okay, so by so if we don't have a cure by 2050, 13.2 million Americans will have this Alzheimer's disease. So this is, I'm not going to go to a great detail about this is the risk factors, what causes Alzheimer's disease, and you can see that what is the main purpose of these things that we have seen that 10 to 15% and almost this is the year of expectancy that Alzheimer's go rising in an alarming rate. And this is the how the Alzheimer's leads, causes, and what are the risk factors. It is genetic. Mostly Alzheimer's are sporadic. It's only 0.1% which is genetic, mostly sporadic either by other stress factors, inflammations, diabetes, and so forth and so on. So this has been published recently by Alzheimer's Drug Discovery 2021 Alzheimer's Disease Facts and Figures, where they shown that if you consider from the last 200 to 219, 2000 to 2019, almost we see that metabolic disease, illnesses, heart attacks, stroke, heart attacks, cancers, the deaths due to these diseases has been decreased by 7.3%, which is great. But unfortunately, deaths due to Alzheimer's disease has increased 145% because so far we don't have any cure. And over 11 million American people has been diagnosed with Alzheimer's or dementias. And the caregivers provided an estimated 15.3 billion hours valued over $257 billion. So there are so many approaches people have looked for. So in which we using various agents, either anti-amyloid, anti-tau, or both combination of anti-amyloid or anti-tau. So these are almost 72%. But this percentage recently came up from the last 5 years that people are starting to look over the other targets where we can do the clinical trials using these either by neuroprotection or used by any acetylcholinesterase activities. So those kind of disease-modifying therapies which are under some of other clinical trials. So what are the symptoms of Alzheimer's disease? Alzheimer's disease is memory loss that affects job and skills, difficulty performing familiar tasks, problem with language, disorientation to time and place, poor and decreased judgment, problems with abstract misplacing things, change in mood or behavior, change in personal. So these are the physically, phenotypically you see an Alzheimer's patient. But how does the Alzheimer cause? So this is the sequence of pathogenic events leading to Alzheimer's disease proposed by amyloid hypothesis. What causes Alzheimer's disease? This amyloid precursor protein, which is an integral membrane protein. It has a normal pathway, which usually everybody, the normal pathways undergoes alpha secretase and gamma secretase cleavage and leads to formation of P3, and where it helps the protein to go into the cytoplasm. But if it undergoes another pathway, which is processing pathway 2, where the beta secretase and gamma secretase enzymes are cleaved, this amyloid precursor protein leads to formation of amyloid beta 40 and 42. Why it's called as amyloid beta 40 and 42? Because when these two enzymes cleave this amyloid precursor proteins, and these are 40 and 42 amino acids, which break down, accumulates, and aggregates. So the question is, what increases risk of getting Alzheimer's disease? Either it is increase in amyloid beta or more A beta 42 formation. Either there's a mutation in this amyloid precursor protein or there's a mutation in beta secretase or gamma secretase cleavage. So another factor which is now alarming in neurodiseases or Alzheimer's disease, that people who have apolipoprotein E allele or mutation, they are more prone to get Alzheimer's disease than the normal ones. So these are the two pathological hallmarks of Alzheimer's disease. So this is the brain. So you can see the healthy brain and you can see the Alzheimer's brain advanced, how there's a shrinkage in the brain. And this is amyloid, this beta amyloid plaques, which are dense deposits of protein and the cellular material that accumulates around the outside or around the nerve cell. But there is also neurofamily tangles, which is the another pathological hallmark in Alzheimer's disease, which are twisted fibers and built inside the nerve cells. So this is the paper which we published in 2010, where that my supervisor, Dr. Charlie Glabe, we found that this amyloid beta, or which is the hallmark in Alzheimer's disease, that all these amyloid proteins, whether there is alpha synuclein is another protein, it aggregates and causes Parkinson disease. PolyQ is another amyloid which aggregates and leads to Huntington disease. Or TDP43 is another protein which aggregates and leads to ALS. So it's a common phenomena on all these amyloid proteins that they are monomeric form. Monomeric form means they have a single unit which undergoes a conformation change and forms these fibrillary oligomers, and their structure changes, and then they form the insoluble fibrils. So it has been seen that monomers are usually alpha helical structure, but fibrils are beta sheet structure. At that time, we discovered one antibody which can recognize these structures, fibrils, this called OC. We named it as OC because UC Irvine is in Orange County. That's why we named it as OC. And then we developed another antibody. What we found that this monomeric protein of any amyloid protein also goes another conformation change, which leads to formation of oligomers. So far, we don't know the structure of these oligomers, and which can be recognized by this another antibody, A11. Then we published a paper with Stanley Prusner, who is a Nobel laureate, who discovered prions. So this may be that these are different amino acids. I don't know what happened. So okay. Just lose of this is okay. Battery. So these are the. So we think that these. I don't think so. It is this battery. Is it the battery? Okay. Yeah. That's it. That's fine now. Okay. That's fine. It doesn't recognize HDMI. Okay, that's fine. Sorry. No, it's fine. I don't know. Okay. Light because it may be the light then. Okay, I think this is the slide which is not picking up. Okay. So anyway, so let me start with various. So how the people are working on developing various therapeutic agents, like some use APP genes to stop production of more APP, or some use copper chelators where they can cleave this A beta monomer, or some use secretase modulators, or some use immunotherapies, which we did at that time, use A beta or tau protein as a vaccine, which can make antibodies and then stop the formation of A beta oligomers or A beta fibrils deposition. So this was the first paper which Elon and Wyeth was the first company in San Francisco when they developed a amyloid beta fibrils and they vaccinated into mice. You can see the unvaccinated mice and vaccinated mice. You can see these round plaques which was stained with 6E10 antibody. 6E10 antibody is a beta specific antibody, which is a pathological hallmark in Alzheimer's disease. You can see this particularly in neocortex, particular entorhinal cortex. These amyloid plaques are reduced in vaccinated ones. So this was the first paper when they published it. So as I spoke earlier, these are the two pathological hallmarks. You can see the difference between normal and Alzheimer's brain, amyloid beta, and neurofibrillary tangles. So I got this slide from John Trojanowski and John Trojanowski and Virginia Lee, who were the pioneers. Unfortunately, John Trojanowski is no more.
Try another thing.
Okay. Yeah. Okay. Okay. So this is the slide which I got from them, and they showed that neurofibrillary tangles, which is the second pathological hallmark in Alzheimer's disease. So because tau proteins, which aggregates and form the neurofibrillary tangle, tau proteins, which normally they stabilize microtubule in brain cells, but they undergo abnormal chemical changes and assemble into spirals, which is called as paired helical filaments, thus creating these tangles and thus disturbs functions and leads to cell death. So this Alzheimer's disease, it's both genetic as well as sporadic. So mostly genetic is only 0.1%, which is familial form, but 99.9% is sporadic. Either it is mutation in the APP presenting genes, which increase the production of A beta 42. The best known risk factor is the inheritance of an ApoE4, which I told you earlier. Between 40 to 80% of the patients with AD possess at least one ApoE allele. Therefore, ApoE allele increase the risk of the disease. Now, these amyloid plaques, which forms inside the brain by A beta accumulation, are neurofibrillary tangles, which is due to tau aggregation or hyperphosphorylation of tau. So these are different amyloid plaques which you can see in the Alzheimer's disease patients. Some are diffuse, some are core plaques, some are neuritic plaques. We also call them dense core plaques. So these are the different structures of these neurofibrillary tangles. Some are flame-shaped, globose-shaped, or some are inside the vessels, which we called as amyloidangiopathy. So far as far the biomarkers of AD progression of pathology is concerned right now, what we have to diagnose these patients Alzheimer's disease. We do structural MRI, we do fluorodeoxyglucose positron emission tomography measurements, or we do febrile A beta PET measurements, or we take CSF from the patient and measure the levels of A beta 42 tau or phospho tau. So these are the So we can use the information from multiple sources to improve the diagnosis and assess the treatment. So neuronal activity, various biomarkers, cognitive reserves, brain atrophies, cognitive functional profiles, genetic profile, or plaque load by using Pittsburgh compound, which detects A beta plaques in the brain. And this was the first studies by Ilan and Wyeth. So when they use the vaccination, that okay, they start treating patients who were suffering from Alzheimer's disease with AN1792. Basically, AN1792 was A beta fibrin, and they started treating the patients. And you can see in the parietal neocortex of non-immunized patient at comparable stage of AD, and with the same patient, like the same patient in the brain parietal neocortex immunized with AD patient in Elan's AN1790. You can see a huge reduction in amyloid plaques. But unfortunately, in phase 3, this whole clinical trials was halted because 6% of patients suffered from meningoencephalitis or other autoimmune complications. So the question came up, this, no doubt you improve cognition, you get reduction in amyloid plaques and tangles, but there's a lot of inflammatory side effects. So we thought, can we produce an effective immune response? Can we produce a peptide or antibody which can produce good immune response and also lowers other inflammatory side effects, autoimmune response, and by restricting the immune response to pathological specific epitopes. So at that time, we screened 30,000 peptides, and we came up with one peptide. So we designed this peptide in the lab, so we named a 3A peptide oligomer antigen. And it gives exactly the same immune response as A beta oligomer. So what we see that is, you can see that this, when we inject it into the mice and we collect the sera from the mice and probe it with this sera where the antibodies against only oligomers, not also only to the A beta 40 oligomer, but also to the calcitonin, insulin, polyQ, not with fibrils or monomer. So this 3A peptide, we started it. It was 20 amino acids long, non-human. It was not showing any sequence homology with the human body, and this aggregates, and we started injecting mice. We use TG2576 mice, and we use vaccine these mice with different antigens. So, and also our antigen 3A oligomer. We injected A beta fibrils, A beta oligomers, IAPP controls via non. So you can see. So this work was done by Hilda. This whole work. So we analyzed it and found that within 8 days, you can see that not only the 3A oligomer, but all the peptides show significant improvement in escape latency. So this Morris water maze is a hippocampal dependent task. And we also did the objective cognition memory. You can see the difference that they improved by vaccinating injecting these peptides, these mice. TG2576 was the first mice model of Alzheimer's disease. They have the mutation at APP, amyloid precursor protein, and also the presenilin 1, but they don't have tau pathology. So then passive inhibitor avoidance, which is a amygdala dependent task, we found that these peptides show significant improvement in amygdala dependent task as well. Then we sacrificed these mice and stained with 6E10 antibody. You can see these 6E10 antibody is an A beta sequence specific antibody. It recognizes A. You can see these controls which were not injected, both hippocampus and cortex. You can see the vaccination with A beta oligomer, IAPP, A beta fibril 3A peptide oligomer. All these peptides shows a significant reduction in amyloid pathology, both in cortex and hippocampus. Then we looked over to the inflammation. So inflammatory pathways, mostly microglia and astrocytes, are the components which usually engulf these amyloid plaques. But in Alzheimer's disease, both microglia and astrocytes gets activated where they lose the power of phagocytosis, where they lose the power of degrading those amyloid plaques and neurofabric tanks. So that's the inflammation. So what we found that these mice which were vaccinated with 3A peptide, when we stain their brains with GFAP, GFAP is an antibody which stains astrocytes. So you can see both hippocampus and cortex that the these activated astrocytes were reduced. That means their phagocytic power has increased. So then taking the same peptide, we got this triple transgenic model from, at that time, Dr. Hilda was working on Frank Lafferla's lab, who discovered this model. We got the mice from them and used the same, because the difference between this mice and the previous mice is that these mice has not only the A beta pathology or amyloid pathology, but these mice has also tau pathology as well. They form neurofibril tangles because they have triple. The reason is that's why they are called triple transgenic. They have APP, PS1, and tau mutation. So we use the same peptide injected in these mice and see, because we see that this 3A peptide reduces amyloid plaques. What about the tau pathology, which is this another pathological hallmark in Alzheimer's disease? So when we injected it after for 8 to 10 months, and then we looked at that, so you can see that vaccination with 3A peptide reduces tau pathology. So we use A beta oligomer as well, IAPP, 3A beta fiber. 3A oligomer was our main target because that's what we discovered this. We synthesized this peptide in the lab and this non-human. People have used it before A beta oligomer vaccination, IAPP, A beta fiber, but all of them was showing a huge side effects. So you can see, so these are the this is the neurofibril tank. You can see the tau pathology here, and you can see in the CA2, CA3, CA4. And you can see here, and this was significantly reduced in mice these triple transgenic treated with 3A. This is the quantification data. So then this was so far I showed you the active immunization. What about the passive immunization? What about if we can inject antibodies? So in the lab, we have discovered various antibodies. I think this was a short project in which we took those anti-oligomeric monoclonal antibodies and injected into triple transgenic mice model, which has both A beta and tau pathology. And what we found that anti-oligomeric and monoclonal antibodies improve cognitive function by reducing A beta deposition and tau pathology in these triple transgenic mice. I think this is the behavior data which Hilda has done so as well. So we can see we gave only 1, 2, 3, 4, 5, 6 doses, and then so we use A11, 204, 205. These were polyclonal and monoclonal oligomer specific antibody, and what we found that when we vaccinate these mice, these antibodies, you can see a significant improvement in latency and all these. And this was the first paper, and you can see that passive vaccination decreases total. You can see these amyloid plaques here, and you can see a significant reduction in amyloid plaques in these mice, triple transgenic mice. They were old mice. Pathology was already developed, so we reversed the pathology. We reduced these pathology amyloid plaque. Now the question came up then, what about the soluble and insoluble A beta 40 and 40? So what we found that vaccination with these oligomer specific antibodies, A11, 204, significantly reduced both soluble and insoluble A beta 40 and 42. Now question comes, so because these triple transgenic mice has both A beta and tau, what about the tau? So what we found that antibodies against this oligomeric A beta not only decreases amyloid plaques, but also decrease hyperphosphorylated tau. So this was the first paper in the world which shows that if you use A beta oligomer antibodies, not only it reduces amyloid plaques, but it also reduces hyperphosphorylated tau. So this was the reason I got my green card, because of this paper. And then in 2014, we discovered this was one of the major change in the Alzheimer's field at that time, where we found that people were knowing that there's always extracellular amyloids. But what we found that intracellular amyloid and the neuronal origin of Alzheimer's neurotic plaques. It was in 2014, we published this paper in Neurobiology of Disease. So what we discovered an antibody which was known as M78. It was a fibril-specific antibody. It recognizes fibril-associated epitopes found in A beta, alpha-synuclein, and it was a confirmation antibody. You can see it recognizes the different structures of islet amyloid polypeptide. Islet amyloid polypeptide has an important role in diabetes. Islet amyloid polypeptide aggregates and leads to type 2 diabetes as well. Alpha-synuclein aggregation leads to Parkinson's disease, and A beta 42 aggregation leads to Alzheimer's disease. So this was the antibody which we discovered. And what we found, we used triple transgenic mice at 12 months, where we showed that at 6 months of age, 9 months of age, 12 months of age, and 14. So this particular M78 antibody, particularly if you see the 12 months, these are the nuclear stains, small stains, which are 8 to 10 microns. But at 14 months of these mice, this antibody M78 recognizes amyloid plaques, which we have never seen before. Then we thought, okay, people have used different antibodies. People have used 4G8, 6E10, and M78. So we took the triple transgenic mice of 14 months age and took their brains and stained them with 4G8, M78, 6E10. M78 was giving us totally different kind of plaques, which people never have shown. So then we did some confocal microscopy. So what we found is, if you see, we took the different ages of triple transgenic mice. This is 3 months, 10 months, 12 months, 14 months, and 14 months wild type mice. So we used two different antibodies. We used 6E10, which is an A beta specific antibody, and M78, which is a confirmation new antibody which we discovered. So if you see, green is 6E10, and this blue is it's a nuclear stain by DAPI we use in the control. If you see in 3 months, you can see a perinuclear staining of 6E10 antibody, which is green, but there is no red staining, no M78. But at 10 months, if you see the red staining, you can see here that this was M78 antibody, which was perinuclear, but it was co-localized with 6E10. But at 12 months of age, when we stain this mice brains with M78, you can see a nuclear staining here, and 6E10 staining, which is green staining, is around. But at 14 months, you can see these antibody M78, these are amyloid plaques in red, and you hardly see any 6E10 staining. So this was a new antibody which was showing a new type of amyloid plaques, and 14 months we didn't see any, because these are wild type mice, they don't have any pathology. So then we did a triple stain, so you can see a DAPI staining. We use triple immunofluorescence, M78, and this is APP, amyloid precursor protein, C-terminal fragment. If you see, so these arrows shows that there's a neurotic plaque where this M78 antibody merge co-localized with this APP C-terminal fragment. And we also use neuronal marker, which is NUN, where you can find this here, that M78 and NUN co-localized together. So this was the first thing that what we say, that the people who say dystrophic neuritis in the brain is actually A beta. It's intracellular A beta. You can see a co-localization of M78 in these dystrophic neuritis. So dystrophic, we say that this is a new type of amyloid plaques which we have discovered. So you can see that at 14 months in triple transgenic, this M78 unique plaque, they have a thioflavin. Thioflavin S is a dye which is used to test these amyloid plaques, but it's also carcinogenic. It binds to amyloid fibrils. And at 12 months, you can see these M78 plus nuclei co-localized with this tunnel labeling. Then we took the brain. So these are the brains of the Alzheimer's disease patients, Brodmann's area, you can see the M78 antibody. It was correlated with the, it shows a good correlation with the intermediate stage of the amyloid plaque load, you can see. And also you can see it, we use different neuronal markers. We see that M78 and these myelin oligodendrocyte glycoprotein is a glycoprotein. It stains oligodendrocytes. You can see GFAP marker, they co-localize it. And you can see the here 6E10 and this M78 is around. And this is the dense core plaque, or we can call this dystrophic neurites, where you can see that these M78 APPC terminal fragment, they completely co-localize. So we say that in previously books were saying in Alzheimer's dystrophic neurites, we found that these are not dystrophic neurites. This is actually a new kind of amyloid protein, which is called as intracellular amyloid, which can be detected by M78 antibody. So this antibody right now not only detects this amyloid, intracellular amyloid, but also involved in lysosomal storage diseases. And Epitomics is selling this antibody now. And this was the whole conclusion of this, that we see that in triple transgenic, where this M78 antibody, positive nuclei observed, but eventually at 14 months, they disappear and stains plaques, and those plaques are totally different from what regular antibody, 6E10, 4G8 or APP antibodies stain. So this was a novel type of amyloid aggregation state where we found the accumulation of nucleus even before formation of amyloid plaques, or even before any cognitive deficit in mice were seen, that we found this new type of amyloid aggregation which involves loss of nuclear DNA as well. So then I went to NYU, where I worked with Tom Wisniewski as a scientist. So we started working on, so we took A beta peptides, we took 3A peptides, we took A beta antibodies. Let us see if we take phosphorylated tau peptide and inject in these triple transgenic models, what happens? So because they have the pathology, they have A beta pathology, they have tau pathology, and we want to see the effect of this particular peptide. It's called as phosphor 37948, 396404. We want to see the effect of this phosphorylated tau peptide in these mice. We just gave 4 injections. And what we found that this was tau immunization. It prevents phospho-tau histopathology, which we use a paired helical filament 1 antibody, which was discovered by Peter Davis. You can see in controls, and you can see in vaccinated one. You can see in the specular variable. And we quantified in both males and females. We see that tau immunization prevents phospho-tau histopathology. And then we looked out at the oligomeric specific tau pathology, which is the MC1. You can see here, particularly CA1, CA2, CA3 region. You can see a significant reduction in these triple transgenic mice injected with phospho-tau, while as you can see the control, how much is the reduction. So we looked out the various soluble and insoluble tau as well, and we found that this tau immunization reduces both soluble and insoluble phospho-tau. And then we looked at this tau immunization not only reduces the phospho-tau, but it also reduces the amyloid beta. When we stain these mice with both 6E10 and 4G, you can see the subiculum area of these mice brain is a control, and you can see the vaccinated ones, this peptide. And this is the quantification. You can see that in both males and females, amyloid beta plaques were significantly reduces in this mice injected with tau protein as compared to the control mice. So as I said that earlier, that inflammation is one of the major first step which leads to accumulation of amyloid beta or accumulation of neurofibril tangles. So we want to see, because in addition to A beta and tau, there is microgliosis and astrogliosis. That means activation of microglia or activation of astrocytes. So we use an IBA1 marker, which is for the microglia. You can see in triple transgenic control mice, non-vaccinated. You can see the spiculum region, and you can see the triple transgenic mice vaccinated with this phospho tau was significantly reduced. And we also found that tau immunization reduces microgliosis in these triple transgenic mice as well. So this was the, so then also one of the major side effect is in this, whether you do active immunization, whether you do passive immunization, is microhemorrhage or meningoencephalitis or any other complication. So you can see in case of triple transgenic mice, with this wild type control, this is TG, 3xTG, and this is wild type phosphotype, this is triple transgenic phosphate, was significantly reduced these microglia. So this was the whole summary what we have done there. This therapy reduces histopathological tau aggregates by 70 to 74% overall compared to triple transgenic control. Further, A beta was reduced. These benefits were associated with reduction in both activation of microglia and microhemorrhage as well. So this was a prophylactic study at that time, and we like to be, this prophylactic therapy will likely be required, which could be assessed first in individuals which has a genetic mutation that are known to cause AD or related tauopathy. However, because the extent of tau pathology correlates much better with cognitive deficits than A beta burden, tau immunotherapies are likely to provide clinical benefits at later stage of AD than treatment that directly targets A beta. So this is the question. So, but so far, we don't have any therapeutic modality for treating Alzheimer's disease. So what is the basic consequences? Because whenever any Alzheimer's patient goes to the neurologist, he does the PET scan and then do mini mental score examination. But at that time, the patient is at the last stage of Alzheimer's disease. So is it possible? Can we detect Alzheimer's early, way before at least people who are 40 years of age will know, oh, we have a biomarker, or we can diagnose Alzheimer's much earlier as compared to. So that's why then after that I joined Johnson & Johnson. So when as a principal chief scientist, so what we named as the dream compounds. Is it possible we can use some probes? Instead of using a brain, we can use eye as a retina, as a window for the brain. Utilization for probes for retinal imaging to diagnose AD and other neurodegenerative diseases, and also to monitor disease activity. And we were thinking it may be and it is currently in the clinical trials phase 3. It will be the first diagnostic biomarker for early detection of Alzheimer's disease and other neurodegenerative diseases. So instead of Alzheimer's patients are who thinks he will go to Alzheimer's will go to an ophthalmologist, he can do an eye drop instead of going neurologist, having a PET scan. And in United States, PET scan is pretty costly. So this is cerebral amyloidangiopathy, is another neurological condition. It's a severe Alzheimer's disease. CAA is a condition that causes amyloid proteins to build up in the walls of blood vessels in the brain. I don't want to go to this. I already showed this. So this is the overall technology we have. We want to make the retina as a window and use this retina to diagnose the brain. So what we did, so we manufactured a fluorophore. We made a fluorophore, and what we did, we injected into the mice of APP Swedish mice by tail, and within 2 minutes you can see the amyloid beta in the eyes. And this was a wild type control. Within 2 minutes you can, and when we sacrifice these mice and we stain with 6E10 antibody, you can see the retina, and wild type there we don't see such staining. You can see this 6E10 and this compound which we injected through tail, and after sacrifice they are completely co-localized. So this was the first investigation where you can detect amyloid beta in the retina using a fluorophore. And this is, we did the same thing. What we did, we took the familial Alzheimer's brain retinas, which we got from Biogen at that time. You can see we use OC antibody, which recognizes amyloid beta, fibril pharmacology. You can see the complete co-localization of this compound. So that means this fluorophore not only detects amyloid beta in the retina, but also in the brain, which gave us a quite good confidence. Okay, so this is good. So you can see this brain compound in the human brain. They completely co-localize with this antibody. And also not only, but also in case of cerebral amyloidangiopathy. And we wrote a grant to NIH to use these fluorophores to diagnose cerebral amyloidangiopathy, alpha-synuclein aggregation, Parkinson's disease, and Alzheimer's. And we got the grants. And this paper we have submitted in last 2 months in Nature Medicine, where we see that this DREAM compound and OC fibropharmac not only outside but also inside the vessels. We can see this compound completely co-localized with this antibody. And this is how it is. You can see these, they completely co-localize it. So if it will be, it is currently in the clinical trials. So these compounds, DREAM compounds, binds to match 10-score plaques and cortical plaques present in brains. And these compounds also detect amyloid beta in retina. And these compounds will be the first diagnostic biomarker where you can detect Alzheimer's disease very early. But also not only Alzheimer's disease, but also other neurodegenerative disease like alpha-synuclein or other alpha-synuclein, Parkinson's disease, Huntington disease, so various others. So this will be the first. So this is under clinical trials now. So this is right now what we are doing. So we use it. I'm not going to show the whole data, but I'm going to show you the conclusion because this is still proprietary. It is still confidential. It's phase 2. So what we are doing that besides so far from the last 40 years, since Elias Alzheimer discovered the amyloid plaques, from 1950 to up to this time, we don't have a cure for Alzheimer's disease. So I don't think so A beta therapy or tau therapy can work. I don't know, Dr. Hilda may have different suggestions, but I don't think so it can work. There should be other alternative methods, like via through inflammatory pathways, neuroprotection, BDNF, or any other. Like, for example, in currently the medicines which are given to Alzheimer's patients, there are 2 medicines. One is acetylcholinesterase inhibitor, which is called a donepezil chloride, and another is memantine. And Dr. Hilda has a paper on memantine, but memantine has also side effects. It reduces insoluble A beta level, but it increases soluble A beta level. She has published that. So far, those are conservative medicine. Conservative medicines means those are just like giving an insulin to a diabetic patient after a certain time, then give again. So they improve cognitive deficit, but doesn't reduce the tau pathology or amyloid beta. So we thought, let's do a different thing. So what we did, we galectin-3, we have found in patients, in mice particularly, first mice, that in Alzheimer's mice, not only the amyloid beta and neurofibrillary tangle, but there's also overexpression of galectin-3 as well. So this is, galectin-3 is, you can read it, but I'm not going to show you the whole data about it. So, but I can show you one piece of data here. You can see it here. So this is the wild type mice, galectin-3, and this is A beta, this is, and you can see APPPS1 mice, which is a transgenic mice model. You can see the A beta level stains with 6E10, and this is the galectin-3 stained with galectin-3 antibody. You can see that in these mice, galectin-3 is highly overexpressed in APP, and you can see this, and you can see that aggregation when you do the different human recombinant galectin-3 with A beta. So galectin-3 intrinsically promotes amyloid beta. So I have to skip these things, but I will show you one part. So this is the thing, what we discovered. that we took the 10 months old APP Swedish mice. We did the behavior before we tested them, and then do a just 2 weeks treatment, 4 doses, 10 milligrams of per kg. That means per week, 2 doses. And then after that, this was before doing vaccinating these mice, this galectin-3 antibody, and this is after. And MOPC2, it's an ISO control antibody. You can see before and after. You can see this TB006, or we can say TB0, show a significant improvement in Morris water maze or hippocampal dependent task. And then we use both models. We use 5x FAD mice, which is a Bob Baser's mice model in which they have a more A beta pathology, because the why it's called 5xAD, because it has 5 mutations. It has APP, PS1, London, Swedish, Iowa mutation. And while this has 2 mutations. You can see this isocontrol, which is a control antibody, and this is our antibody, which shows a significant reduction in amyloid plaques. So I'm not going to this data. So overall, I will tell you one thing. So this is what are the current ones people are working on. So Biogen's, Roche, SI, Eli Lilly, Lundbeck, AC Immune, Jensen, FV, Lilly, Elector. So these are the things what they are noticing. You can see one thing is common in all. What is that? Amyloid-related imaging abnormalities, amyloid-related imaging encephalopathies. And you can see the cognitive deficits, efficacy, microhemorrhage. You see this part. All these so far, what these guys have done, everybody is not having, either they didn't show the data or on microhemorrhage or other side effects when they use their own peptide, but whoever shows, they were showing a huge amount of microhemorrhage data. So this we discovered that on last year, this was approved by, we filed an IND approved by FDA until now, July 2022. So we didn't find any autoimmune complications and no side effects in healthy human volunteers. And 278 moderate and severe diagnosed human subjects were dosed with this single ascending dose of 10 milligram per kg of this galectin-3 antibody. Currently, this clinical trial is still going on in 25 sites all over the United States. And this whole work is this is my current group, which are working on these different projects, particularly Jenny Johnson and Luda, they work on Alzheimer's disease. Sangmi Lee works on amyloid ALS, and Nancy and Thomas, they work on Parkinson's disease. And we started recently a new disease that's called as age-related macular degeneration, where you can see a deposition of A beta in the eyes. And that's started by Barbara. And she's working on various tauopathies like PSP, Pick's disease, and cortical. These are 2 researchers who are working. And finally, this is what we were before. And this is New York, this is Disneyland, Irvine. And I used to have various grants and supported by NIH, and my mentors who were there, Charlie, Einer, and that's it. And I will be happy to answer any question. Thank you. Yeah. Yeah. Yeah, it's a candidate confirmation antibody.
It could have an effect also in reducing, in lowering the levels of alpha-synuclein or other proteins.
Yeah, that's what we did. Yeah, that's what we did. Yeah.
So that's fantastic because it will have like a wide range of applications.
Yeah, it is currently, it is epitomic, millipore, and sigma. They sell this antibody, not for the various neurodegenerative diseases, but also for lysosomal storage disease, because it dissolves aggregation of various proteins. This is a conformation-specific monoclonal fiber-specific antibody.
I want to know your opinion about like
Simerson's work about injecting antibodies to phosphorylations of tau, specific tau phosphorylation antibodies, like immunotherapy instead of amyloid or neuronal.
So people are using various approaches, either using active immunization. Einer is using this. Einer was the first one who actually started using tau antibodies or tau peptide is used as an immunotherapy for Alzheimer's disease. But the problem comes that, as I told you, that major side effects. So because all these proteins, whether you use A beta or whether you use tau, phospho, tau, or any other antibody, they go to immunologically, they go to Th1 pathway. Th1 pathway means they activate T cells and then activate plasma cells, which then secrete B cells. And then these B cells not only secrete, they secrete various inflammatory cytokines. That's the problem. So if we have another approach, that's why we came up with 3A peptide. 3A peptide is a short peptide, non-human. It doesn't go to the regular pathway, Th1 pathway. It goes to Th2 pathway. That means it activates directly B cells instead of going to T cells. So that's the approach we do. But unfortunately, this peptide took by GlaxoSmithKline, GSK1. But it didn't cross much efficacy in the blood-brain barrier. That was one problem. Thank you. Yeah.
I have two very basic questions.
Yeah.
The first one is related to the mechanism by the antibodies can cross the blood-brain barrier, because usually the antibodies cannot cross that barrier.
Yeah, that's a question which everybody asks. That's why. So even 0.1% antibody could go, it's showing efficacy. Now Genentech and Denali, they have discovered a transporter. What they do, they find the transferrin with the antibody, which they publish that in Nature, which increases the penetrance or permeability of antibody, which is actually a good idea. But now the mechanism of AI, artificial intelligence, that they want to know, they can screen the antibodies, which can probably cause the burden, like for the adenosine Biogen's antibody, or Elector STROM2 antibody, or SI's Lenacapavir antibody. Only 0.41%, but it shows effects. The problem comes the side effects. It's not now the people show that how much antibody goes into the brain. So three main aspects are that. Whether you have your product, whether it's antibody or protein, have the potential to reduce amyloid plaque and damage. Not only that, cognitively, but microhemorrhage and other amyloid-related imaging abnormalities. One of the main concerns, safety. You want to see whether this immunization, whether you do any antibody or protein, is safe for you. That's why if you see adenosine, 20% patients suffer from encephalitis. Yale guys published paper last month. Patient who died, they screened her brain, tau pathology was alarmed in the brain. And they are currently now. Oh, this is the problem. I don't think so. A beta tau peptides as a question is going to work. People have used it for the last 50 years. It has been how many years? 73 years since Elias Alzheimer's discovered Alzheimer's. We are nowhere. Yeah. And it is increasing. It's increasing. Deaths. So last year it was 6th cause. Now this year it's 4th cause of death. And I actually increase funding. The problem is here. Adenosine, it was an accelerated approval. In order to give hope to the people, oh, it's an accelerated approval, we can approve. But Medicare totally rejected it. We are not going to say that because it's a plot. This is the one main question.
And the other question, very basic. So what is the normal function of the gut-free physiology?
It has a normal function. Basically, it's a galactopeptide. It's a glycoprotein, which basically helps in glycosylations as well. But it's but it's most associated with microglia. So what this antibody does is not only reduce the activation of microglia, but also increase the phagocytic activity. It's not a direct target, it's an indirect target. We are not targeting A beta, we are targeting microglia, which can target it. Go to our website, you can see there, this is how we have shown that. So because this antibody reduces the activation of microglia, indirectly increase. I have put the video in the website, and that increases the phagocytic activity of microglia, which later engulfs these amyloid plaques without side. That's our approach.
Thank you.
Yeah.
I have a question about your approach to detect, to early detect.
Early detection. Yeah.
How differentiate between is Alzheimer's disease or another neurological disease when you are detecting, I don't know, inflammation, vascular inflammation? What is that detecting with brain compounds?
Yeah, so that's we have different fluorophore. That's what is one thing. So like alpha-synuclein aggregates, like neon aggregates, like A beta aggregates. How you are going to differentiate if you detect in the eyes? So for that purpose, so we know by shape. So it has been shown, one of the person who is from Cedar Sinai, her name is Maya Cordova. She was the first person who showed that amyloids in the retina and amyloids in the brain, they're almost similar. And alpha-synuclein has a different structure. Alpha-synuclein has totally different. Yes, now it will be difficult for an ophthalmologist when he can put an eye drop and do a retinal scan, whether it is alpha-synuclein or whether it is A beta, whether he will have Alzheimer's. For that, we have basically just designed some specific probes for specific diseases, like fluoroprobe. So basically, why? How we came up with this idea. So this idea, this fluorophore, it doesn't fluorescence, it doesn't emit light. But when it binds to A beta, it emits fluorescence 500 times. So now we want to take this fluorophore and synthesize different fluorophores so that they can bind to different amyloid, specific amyloid peptides. That's the plan.
What about the amount of the beta amyloid do you need to detect that the beta amyloid to? I don't know.
So now there are blood biomarkers like recently C2N is a company. You can take the blood and they can detect the amyloid beta levels in the plasma. There you go. That's also one of them. But the use of these fluorophores we want to test earlier, as before it goes into the brain, before even patient goes to cognitive deficit, before his mini mental score examination lower, we want to detect this pathology early. That was our main target. Yeah.
Well, first of all, thank you to be here. It's more interesting. And my question is, so crazy, it's crazy. I like when you talk about the immunization. My question is, do you think the Alzheimer's disease disappear in the future or reduce the cases in the world?
It's $1 billion question. I wish I would have answered about it. As far as its progression is concerned, it's increasing by alarming rate. And last year, Biogen's antibody, they gave a hope, but it didn't came through. So this time, Eisai has another antibody, it's called lecanemab. They are showing it may be a progress, but I still doubt it. But I think best solution would be change the social setup as far as America is concerned. I don't know about Mexico, but as far as America is concerned, change the social setup. Nutrition, eat healthy food. Social setup means, like for example, India. I don't know whether hardly any person diagnosed with Alzheimer's because 3 generations lives under the same roof, and the older person don't have to go for work. He just has to enjoy his life and his son or daughter take care. But in the United States, they prefer dad. They prefer son, they prefer father. So that's the problem. The social setup is one of the big. It leads settlers. It leads formation of more reactive oxygen species, leads to diabetes, which leads to Alzheimer's. But I hope, as far as avian or tau, but there are other things, other people are working on various using neuroprotection, BDNF, or some other neurotransmitters. They can help. I hope it should work. Our should work. What we hope. Yeah.
Was like your seminar story, great data. You found that colonization produced a very nice improvement in the pathology of Alzheimer's disease, whereas microglosis,
Yeah.
So, what do you think? I mean, I have two questions. The first one is, did that correlate with clinical improvement? In the
Tau has a more efficacy because tau is a normal protein. It's a tubulin associated with a microtubule stabilizing. It has a function. When it hyperphosphate, then it leads formation of neurofunction. Whereas A beta does have a function. A beta is produced from protein, but A beta acts as a seal, which leads more. So tau immunotherapy may have little bit better impact on Alzheimer's disease as compared to.
So my second question is, related to this. What's your personal opinion about the contribution of A beta theory versus the tau theory? I mean, there are, lots of one.
See,
There is a more.
so I have worked. I do that. We have worked with those persons who have made breakthroughs in Alzheimer's. Like Lafferla was first person who discovered tryptophan. Charlie Day was the first person who discovered A beta oligomer. Before that, everybody was saying that A beta fibers are toxic. But he published a paper in Science and showed no, before fibers, there's a one structure which is very unstable, which is more toxic. Those are oligomers. Those are higher molecular than lower. Regarding A beta things, we don't know the structure of A beta. That's a problem. If we know it, we can start targeting. Another thing is that keeping the consideration of amyloid precursor protein or presenilin 1 or presenilin 2 genes in consideration. I think interference RNA would be a good so that you can know by genetically how this works. What are the genes are involved? We don't know how many genes are involved. We are saying lysosomes and no lysosomal part. 2 days ago, there was a paper published by Dave Halsman, and TREM2 is involved. Now another came, PISO1 is involved. How many genes are involved? What are you targeting? Are you targeting A beta or are you targeting tau? How much you are targeting? What particular conformation you are targeting? Since still people don't have an idea. And particularly, it's just like who comes first, whether it is chicken or whether it's egg. Whether it's A beta or it's tau. Some people say tau, some people say A beta. Still, one of the questions which is still a billion-dollar question in Alzheimer's field. There are so many things. I think that my personal experience from the last 13 to 14 years, what we have done that we don't think that A-beta immunotherapy would be a better choice for Alzheimer's. So for that, we should know the possible mechanisms. We should know all the pathways. I think genetically, RNA, or using any other inflammatory pathways would be a better approach rather than what the
Can you comment about these, all these press hype about monoclonal antibodies?
yeah, lecanemab.
Yeah.
If you have money, you can do anything. That's my take. I asked them, so in the CTAD conference, Clinical Trials Alzheimer's meeting last week, which was in San Francisco, because Eastside was the gold member of this clinical trial, and they paid $1.5 million to the clinical trials meeting. And when they gave a talk, I was the first person who asked the question that, can you please tell me this lecanemab, why it binds? Which particular size it binds? So they say it is protofibril. Protofibril is above mixture of oligomers and fibers. So it's 50 kilodalton protein. But the problem is that more toxic ones are dimers, trimers, tetramers. Those are the toxic ones. What happens to those? So it is just investing money. Richer becomes richer. That's it.
So you're skeptical about it?
No, I am skeptical. I am 100% sure. I am skeptical. Yeah, okay, it shows benefit, it works. But let us see how many patients have died. So Biogen have done, they didn't show the whole data. And lecanemab antibody is much more promising as compared to Biogen. But still, cards have yet to be open. This phase 3 is going on. Let us see what they come up with. So amyloid-related imaging abnormalities, amyloid-related imaging encephalopathies are the main important things. How safe the drug is. These things are still to be validated. Let us see what they will come to the next year. But still, as far as the current atmosphere in Alzheimer's are concerned, people have hope. Something will come. But to be honest, my experience is, we did, as far as immunotherapy is concerned, we didn't make a major breakthrough. We are still there. But one thing important, regarding the biomarker, we made a huge impact. We can detect the beta in plasma, which never detected. We have different tools for diagnosing Alzheimer's. We have different tau antibodies, phospho tau 217, 181, 231. Those are phenomenal biomarkers. At this point, we make a good progress, but immunotherapy-wise, no. Biomarker-wise, yes, I agree with that point, because we have worked on it. We saw it. Yes, these biomarkers are validated, particularly C2N, C2N, PSI, Contrarex, Roche. Biomarker-wise, we are in a good position. But therapy-wise, we are still at the same since 1950.
That is disappointing because what's the point of making a early diagnosis if you can't do anything?
Yeah, exactly. But at least you can assess the patient very quickly what is Alzheimer's. And even in some cases, you may not need to go positive on emission. And in the United States, it's very costly. Mostly health insurance doesn't cover it. It costs $8,000. Yeah.
Original imaging can be used as a biomarker to diagnose Alzheimer's disease, no matter the stage of the pathology.
That's what we are thinking. That's why the second clinical trials, phase 2 second clinical trial. But we have to screen the probes which are selective for different amyloid proteins involved in different neurodegenerative.
What we have to see in our patients to say that
we want that instead of going to a neurologist, patient can go to an ophthalmologist where he can use an eye drop, put in the eye, put a dilate, and then do a retinal imaging scan. So you may have a possibility of Alzheimer's disease. So you can take some herbal medicine, or some take some nutrients, curcumin, or some resveratrol. There are so many things which can stop progression of Alzheimer's. Yeah.
What is your opinion about to use non-pharmacological treatment like
I love it.
exercise or
I love
maybe, I don't know,
I am with it.
polyphenols, I don't know.
Yeah, I am with it.
Do
I
you
published a
think
couple of them.
that's gonna work?
Yeah, resveratrol, biophenols, fish oil, omega-3 fatty acids. So
For
I have published.
prevention or for early treatment? What is
I
it?
think early treatment. That's why Indians don't have much Alzheimer's because they take curcumin every day. They take curcumin every day. But this is a good approach. Yeah, because the problem with curcumin is you cannot give curcumin, you cannot eat so much because it metabolizes quickly and it leads to curcumin toxicity as well. But omega-3 fatty acids, great. Resveratrol, great. Drink red wine every day.
I had some of
Causes liver problems. Yeah.
Hear more about your opinion of the plasma samples for diagnosis versus the CSF. Here in Mexico, it's costly and very dangerous to obtain this kind of samples.
Yeah, that's why a lot of Alzheimer's patients, they were not cooperating with having the lumbar puncture connected to CSF. CSF is pretty painful. That's why now C2N, Confocal, or Simoa, they started using a plasma test. Take the blood sample, collect the plasma, and see the levels of A beta levels and A42 levels. But you can see the p-tau levels. Now we don't need the CSF as well. This is a quite good progress. That's what I was telling that. As far as the biomarker wise is concerned, we progress a lot as compared to a couple of years ago.
It's possible to get samples of plasma and try to use for the
But it so these guys, they have a particular antibody which can detect A beta in the plasma.
but the levels are so low.
Cells are very so low. Levels are very low. Hardly detect A beta in plasma, but they have a specific antibody. They also comes to C2N laboratories. It's $1 billion company. They can detect A beta in very extremely small levels in plasma, which can be used, and it's FDA approved that you can use this test to detect the A beta levels in plasma.
¿Alguna pregunta? Bueno, pues, doctor, thank you so much for your lecture, and on behalf of the head of our department, Dr. your presentation for this incredible lecture. Thank you so much. Muchas gracias a todos por su asistencia. Este es el último seminario de este año, entonces nos vemos hasta el 2023. Muchísimas gracias por su asistencia. Igualmente en redes sociales. Feliz año. Yeah, sure.